didn't expect everyone to be so quiet all at once. So I figured I'm obligated to say something now as people start rolling in.
Well, I can start. I just wanted to
Oh
just wait and
no,
see.
like literally we've got 8 people, including the 5 of us.
Yeah,
So,
that's
we're
what
in
I
great
kind
shape.
of was noticing.
Yeah. 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 Oh, I guess Pam, we got to talk tomorrow too. I've got another MC request. You're becoming quite popular in these things.
Hey, you've got to make sure that you also get me the soybean guest
Yes.
ASAP,
Yeah, for
or
sure.
or tell me if you want me to
No,
hunt around. But
thank
just
you
tell
for
me,
that. I reached out to Jordan Lamb on that and she hasn't got back to me.
who is it?
Jordan Lamb. She's our attorney, our on-staff attorney,
Oh
and
yeah, yeah,
yeah,
yeah, yeah,
very
yep, yep,
very
yep,
smart gal.
yep.
Um,
Sweet.
yeah.
Yeah, that'd be a good one.
Um, okay, it's starting to get real. We're up to 9. Thanks for your patience, Adam. Steve, we're just getting fired up here. Pam will start her spiel here in a minute. We're letting people roll in. Appreciate you joining early and often. But yeah, Pam, we're the annual meeting is going to be virtual on the 18th,
Okay.
and there's been a few requests to help with that because there's not a corn soy expo and other opportunities for our partners to kind of be in front of people. And I
Mm,
already told them, and I told Rochelle to mention it to you as well, but I not a requirement, but I'd love to give you first right of refusal because of course we would always love to
that's
have
a
you.
that's a Thursday.
I believe so, yes.
Thursday, the 18th of February.
Mhm.
Yeah,
I'll send you the
I'm
details too.
I'm game on. Yep.
Okay,
Just send
cool.
me the details on when you need me.
Yeah, thank you. Thank
And
you.
is it Jordan Lamb? Jordan Lamb, you said you're
Mhm.
thinking is okay.
That's why
Beautiful.
who I'd really like to, just because it's all the political stuff going on right now. I want to do a better job of having people outside of the immediate ag
Right.
world.
No, I get you. There's no conflict with the checkoff.
We're gonna find out.
Okay.
She would be somebody to tell us.
She's a good interview.
Yeah, if she'll do it.
Right. Yeah. Gotcha.
Okay, Tom, welcome.
All right. I can roll if you want.
I just there's a couple of people that I've texted that are trying to join that Eventbrite won't let them join yet.
Interesting. Let me just log in, and I wonder if it's full.
I think they're saying that the event hasn't started yet, according to Eventbrite, so it's not.
Interesting. We are trying to do it a little differently this time. Last time we sent out extra notifications that allowed it just created a lot of confusion because I changed it to 15 minutes earlier. So this time I set everything to allow anybody with the Zoom link to join anytime. And I'm getting a lot of people joining. So I think we are going to get just a mass inflow at 7 then because of Eventbrite. Let me see if I can change that quickly.
Well, in the meantime, why don't I get started welcoming folks in that are on their way into the room? Thanks so much for joining us, everybody that's patiently trying to get logged in. Keep circling back around if it's taking you a moment. My name is Pam Yankee. They call me the fabulous Farm Babe Pam in southern Wisconsin. Here I do TV and radio farm broadcasting for the state of Wisconsin. Happy to be your MC again for the second installment of Beans and Bull. Tonight, we're talking about on-the-farm research, cover crops, as well as soil health. I'll be introducing you to some of our guest speakers here in just a moment, but we appreciate you joining us. And as a matter of fact, because you're early to the virtual conference, we're going to make it worth your while. And we'll be doing this during every one of the Beans and Bull conferences this spring. $50 gift card to Cabela's for the correct answer to one of our very intriguing trivia questions that I'll be bringing to you coming up in just a moment. But again, like we said, we're planning on an hour's worth of conversation tonight about on-farm research, cover crops, and soil health, and maybe making sure everybody's on the same page as far as some of the definitions on things like soil health. So stand by for that. Also, if you are somebody that's joining us for CCA credits, just a pleasant reminder that when it comes to you trying to get the credit for being a part of this virtual discussion, remember that you have to kind of enter your information at the end unless you're using the app. There'll be more conversation about that when Dr. Shawn Conley, our University Extension soybean specialist, steps in. So just a couple quick things as far as warm-ups are concerned. I know that many of you are going to be from the state of Wisconsin, and you're probably going to be very familiar with some of the things that I'm going to share with you, but that's just fine. It's 7 o'clock on a weeknight. We're going into a big game as far as Wisconsin and our Packer fans are concerned. So I thought I'd just remind you about some of the things that you may see in Wisconsin and need to correlate it with the importance in the state. First of all, we're all connected to agriculture in one way, shape, or form, right? Considering that we've got a guest that's going to be on with us from the East Coast, shall we say, Wisconsin has declared one of the products that we are number one in as far as national production, the state fruit. Stop and think for a moment. What state fruit is Wisconsin gorilla in as far as production is concerned? That's coming up in just a moment. Also, some fun things that my assistant pulled for me. How many gallons of sap does it take to make maple syrup? You know, in about a month or so, believe it or not, they'll be tapping trees in Wisconsin trying to put together some of that sweet sugar that we look forward to on pancakes and other things. They tell me that it takes about 40 gallons of sap to make just 1 gallon of syrup. So think about that. Typical harvest for Wisconsin maple syrup producers is only 4 to 6 weeks. And given the weather that we've already had, today was a pretty beautiful day in southern Wisconsin with temperatures near 40 degrees. That window of opportunity for our maple syrup growers might shrink just a little bit. Speaking of shrink, they shrink as we harvest them. I'm talking about our Wisconsin cranberries. We produce 60% of the nation's cranberries, and we have that as our state fruit. So again, if you're kind of new to it all, there's a couple trivia tidbits that you can find out about the state. Again, my name is Pam Yankee. They call me the Fabulous Farm Babe. I'm really happy to welcome you all in to the second installment of Beans and Bull. Tonight, we're talking about on-farm research, also cover crops, and soil health. I'll be introducing you to our guest coming up in just a moment. If you are trying to earn some CCA credits in tonight's conversation, please remember that you should be entering your information, your CCA number, at the end of the seminar. I also understand that Dr. Shawn Conley is going to keep a lot of the big announcements you're probably waiting on, especially if you're from the seed industry, until the tail end of his presentation. So you might want to have a beverage or a snack nearby so that you can hang with us till closer to 8 o'clock tonight. Of course, badgerbean.com is one of the places where you can always connect with upcoming Beans and Bull session. Also coolbean.info, that's Dr. Shawn Conley's site. And of course, he is a lover of the Twitter feed. So make sure that you get signed up for that as well. Talking a little bit more about some of the activities that are going to be going on, I want to remind you about some of the other activities connected with agriculture that you don't want to forget. Midwest Forage Association is going to be having some virtual conferences as well, talking about cover crops tonight. That goes hand in glove with what's going on with Wisconsin's alfalfa and hay production. Please make sure you check out Midwest Forage Association's virtual conference that's going to be held in just a couple of weeks. Also, they are telling me that Commodity Classic, although it's going to be virtual, is going to be very, very interesting. I know today the organizers of Commodity Classic asked if I would basically moderate a panel on the state of farmers' mental health. So they've got a lot of interactive things planned for not just keynote speakers, but for special guests from across the United States. So stay tuned for details on that. If you want to stay tuned for details, I am happy to provide you with those. You can follow me, Fabulous Farm Babe, on Facebook, Instagram, Twitter is Fab Farm Babe. Also, MidwestFarmReport.com is our website where we try to keep up to date on all the news pertinent to Wisconsin agriculture. And of course, if you are interested in listening to something anytime, whether you're up early or up late, you can also stream The Farm 97.7 FM, and it's now Alexa enabled. So just tell her to find the skill for The Farm, and you can also listen to that farm information literally 24 hours a day. So let's talk a little bit about the guests that are going to be with you tonight. First of all, we want to be sure to welcome in Lindsay Chamberlain. She's a graduate student at UW-Madison working with Dr. Conley. She is from Western New York. We were talking football before we kind of all got together. She grew up on a dairy farm, God bless her. She graduated from Cornell in 2017. She's been in Wisconsin working with the bean team ever since. Tonight, she's going to be talking about what she's learned in that time about cover cropping and soil health in soybeans. Then we'll bring in the master, Dr. Conley, Cool Bean. He is, of course, our state soybean and small grains extension specialist, and he's going to talk about the 2020 Wisconsin Soybean Yield Contest results. That's the one I think a lot of you are curious about, including yours truly. He's also going to have some data from on-farm in-field work, benchmarking some real boots on the ground on-farm research. So that is what you've got coming up in just a couple moments. We do want to try to be respectful of your time. So tonight, we are focused in on trivia. And since we're talking about soil health, Farm Babe thought we'd get a little education on earthworms. So now remember, the way this works is if you've got the correct answer to tonight's trivia question, you can email it to farm@midwestfarmreport.com. Simple as that, farm@midwestfarmreport.com. And the correct answers will randomly select a winner for a $50 Cabela's gift card. Now, pass it on to your buddies. I'm going to have a $50 gift card for every one of these Beans and Bulls sessions, and you've got to be an early comer to qualify. So a couple things maybe you didn't think about as far as earthworms. They have no arms, legs, ears, or eyes. Okay, that's not too odd. Also, if you took a look at an acre of land, a healthy acre of land, they estimate probably is home to about 1 million earthworms. Now, here's the trivia question for tonight as we approach the 7 o'clock hour. The largest earthworm ever found was measured 22 feet from nose to the tip of its tail. The largest earthworm ever found. Trivia question, and again, remember, send your answer to farm@midwestfarmreport.com for a chance at that $50 Cabela's gift card. The largest earthworm ever found 22 feet from nose to tail. Where was it found? Farm@midwestfarmreport's the email you want to use. And again, the question, it measured 22 feet from nose to tail, world's largest earthworm. Where was that earthworm discovered? That's keeping the spirit of our cover crop on-farm research soil health conversation that is part of Beans and Bull. Again, my name is Pam Yonke. Thank you so much for joining us. We appreciate you coming in early. That's why the $50 gift card is a part of the bait to get you here. Now, without further ado, let me turn over the program to our friend Lindsay Chamberlain, who tonight is going to focus in on just exactly what's been going on as far as soil health. Everybody has their own definition of that, and Lindsay is going to share with you what soil health really means, how can we manage it, and we're also going to focus in on soil health, cover crops, and cover crops with a soybean rotation in mind. Dr. Shawn Conley will follow with our 2020 Soybean Yield Contest. Have a great evening, everybody. Lindsay, take it away.
Still muted. Thank you, Pam. I can get my slides to switch here. This worked in the tech check.
Yeah, if you have two monitors plugged in, it might be sending you to the other screen.
I switched them last time and I can't remember how I did it.
If you unshare your screen, you should be able to turn it into present mode, and then you'll be able to select the screen, put it in present mode, then share your screen, and it'll ask you to select the other monitor.
Having trouble with the Zoom screen sharing. I'm gonna do something else really quick. I'm sorry about this, everybody.
No problem. People are still joining. So I apologize too. Eventbrite didn't want everybody to join early, even though Zoom was allowing everybody to. So thank you for those that figured out how to get in early. And we made sure that everyone who joined on time at 7 had a chance for the trivia question with Pam Yankee. Emma has posted the email to send your answer into that, and the CCA credit QR code will be presented at the last slide of Shawn's presentation. Again, at the very end, you'll also be asked to present. If you don't have a QR reader, you can post your number and name into the chat at the very end. We will export those and we'll get those entered for CCA credit. Again, that's at the very end of the presentation tonight. After the presentations, we'll also have Emma collecting questions you have, and she will politely interject herself between the two keynote speakers, read any questions posted in the chat. And again, at the end, she'll do the same as well. So if you have any questions, feel free to post them in chat. Otherwise, there'll be a time at the end to ask questions as well. I think we're ready. Do this again? Okay. People are still rolling in, so it worked out just fine.
Great. As if you joined after Pam introduced me, I'm Lindsay Chamberlain. I'm a PhD student working with Dr. Shawn Conley at UW-Madison. And tonight I'm going to just share some of what I've learned over the last couple of years about cover crops and soil health in soybean. And I will be somewhat candid with you. I have some sort of preliminary data to show you and some interesting things. As I'm sure all of you know, cover crops are a multi-use tool with a variety of benefits. It's reducing erosion, weed management, potentially building soil organic matter, and improving soil health. And I have soil health in quotes there as just sort of a nod to the second half of my talk, where I'll try to talk a little bit about the sort of vague definition of soil health and maybe elucidate it a little bit for you. But these also come with a lot of challenges. It's a steep learning curve to figure out how cover crops fit into your rotation. Establishment after soybeans in Wisconsin or corn is really hard just because there's not a lot of growing degree days left. There's not a lot of heat left in the year. Another factor, I think we talk about the time, labor, money aspect of adding to your rotation, but it's also another management decision. It's something else to think about complicating your operation. And I think that's worth considering in terms of adding to the farm stress that already exists. And then finally, there's yield reduction worries. And hopefully I'll be able to dispel that worry a little bit tonight. So I'll be pretty candid with you. The UW Bean team is learning along with you. There is a ton of awesome cover cropping research that's already published and out there. If you're interested in it, I'm going to talk about 2 different projects that the Bean team is working on right now that are not polished and finished yet, but we've learned a lot about cover cropping as we worked on these. So the first one is a planting soybeans green into cereal rye project, and I'm a little less involved in that one, but I'll show you a little bit of data. And the second project is one that I'm more heavily involved in, and that's more of a crop rotation cover cropping systems approach, looking at several different cover cropping options, not just cereal rye. So this first one, this is soybean yield for this planting green study at 5 different sites. We've got just one Wisconsin site in Arlington. Then this is Arlington, Nebraska, Shelby, Nebraska, Lamberton, Minnesota, and Rosemont, Minnesota. The stats were done within each location here. If that, so these being A's and these being A's isn't saying that those are equal. But what I want to draw your attention to, there are 4 treatments: no cover crop control, an early termination timing, an at planting termination timing, and then a late termination. And that late termination, I think, was about 7 days after soybean planting. And looking across, so comparing the letters to the no cover crop control, there was only one instance over all of these cases where there was a reduction in yield compared to the no cover crop control. And that was at Arlington, Wisconsin, and it was just with the it was with the at plant. So that there was a reduced yield with at plant termination timing, but not with the late termination timing. So the point of this is to say that there is going to be some variability, but most of the time from this project at least, there wasn't a yield reduction with soybean that followed a rye cover crop. Whoops, I had an arrow to emphasize it for you. So this is another project that I'm a little more entrenched in, and it is a corn, soybean, wheat rotation study that's in Arlington, Wisconsin. These are the 6 different schemes of rotation. We've got continuous soybean and continuous corn, annually rotated corn and soybean, then corn, soybean, wheat, corn, wheat, soybean, corn silage, wheat, soybean. And these rotations have been in place since 2002 when the study was established, but there's been a split plot of cover crops since 2017. And those cover crops are they're not necessarily dependent on the rotation system, but what the crop is. So after corn or soybeans, we drilled or broadcast, well, we drilled and we broadcast either oat or cereal rye. We did both species both ways. And the broadcast treatment was into the standing soybean. So that happened, or corn, that happened the first week of September-ish, mid-September for corn. It was a little later for corn. So that's kind of simulating a plane flying over. Since these plots were so small, I did it with a yogurt cup. Following wheat, we used either red clover or Berseem clover, and we drilled it after wheat harvest, or we frost seeded. And frost seeding actually happens in March. It's kind of taking advantage of the freeze thaw action that's going on in that early springtime and drawing the seed in. So you might notice, if you're familiar with these species, so oat and Berseem clover are both not winter hardy in Wisconsin. They'll die over the winter, whereas red clover and cereal rye will survive the winter in Wisconsin. So these are just a couple pictures of the most successful cover crops that we had. Successful just meaning that there was a decent amount of growth. This is a red clover that was frost seeded. And then here is a patch of rye. Well, it's a plot of rye that was broadcast seeded. And then we have the drilled treatment of cereal rye next to it. So this is also a difference between a September seeding and an October seeding. And this is the day that these cover crops are sprayed. So that's as much biomass as was there. So to sort of sum this up in terms of yield, this is soybean seed yield for 2018, 19, and 20. So 3 years of cover cropping sort of all put together. And you might notice that these bar graphs, or these box plots, are red and orange, and these ones are blue and green. That's because of the type of cover crop. So these two rotations, you've got the CWS, or corn, wheat, soy rotation, and corn, wheat, soy rotation that's corn silage. And these soybean rotations were following clover cover crops, whereas these were following grass cover crops. This is just preliminary data because of the different species. The statistics have become pretty complicated with the study. But this kind of shows what I would like you to get from it, is that rotation seems to matter a lot more than the cover crop. But there is also quite a few outliers here. And if we split it out by year, we can see that that's really coming from one rotation in 2020. And these, since I was standing in the plots, I know we had some trouble with terminating that red clover and terminating some of the volunteer wheat as well. So just a couple of lessons from this study. Effective termination is really key for limiting impacts on yield from cover crops. So you need to be scouting out there and trying to figure out when to terminate based on how much growth there's been, and that might be different between different years. Secondly, small plot cover cropping research is really difficult. Cover crops have variable establishment, and so establishing a solid cover over a 10 by 30 foot plot is tricky. So for the planting green study, we actually did have considerably bigger plots than for this one. That's just something to keep in mind when you're looking at other research, and also if you're interested in putting some out on your farm, strips for across a whole field might be a lot more useful. Lastly, limited growing degree units after typical corn and soybean harvest in Wisconsin is a problem for cover crop establishment. So if you are looking at trying to put out cover crops after corn or soybean, I would really consider aerial seeding. I think that's a great option in September, or planting a shorter maturity cash crop in order to get those in. We did plant a slightly shorter maturity soybean in this study in order to get both the wheat and the cover crops in a little earlier. All right, so switching gears, I'm managing for soil health, but how do I measure it? The USDA defines soil health poetically, I think, as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. Like I said, I think it's poetic, and it's a good definition of soil health, but it's hard to use that to then manage soil health. What does that really mean on your farm? I think this Venn diagram, I saw Steve Coleman present this information this way once, and I really like thinking about it as this Venn diagram, sort of the crux of physical components, chemical components, and biological components. And in your nutrient management plans, you're probably thinking a lot about the chemical components already, just as something to think about. So soil health really comes at the crux of these things when they all kind of come together and are functioning well. And this is simple in theory, right? We have, for physical components, we have texture and structure of soil. For chemical components, these are likely things that you're already measuring, like pH and the nutrient status. And then biological components, thinking about soil organic matter, you might be measuring that. And then microbial communities and microbial community activity. This is something, I know my dad was excited to bury a pair of white underwear and then dig them up and see how much they were degraded. And that's a test of how active the microbes are. But when we start thinking about lab tests, this list gets really complicated really quickly. A lot of these are really expensive and might not offer you that much information. And to do them all is to conduct a research study, not necessarily something to manage on your farm. But I do want to point out some of these physical components, like thinking about compaction, penetration resistance. Texture is something that you can't necessarily change on your farm, but these might be things that you can go out with a shovel and think about. Chemical components, a lot of them you're already measuring, like I said, pH, nutrient status. But these biological soil health indicators, they're not the most important part of soil health necessarily, but a lot of the soil health indicators, the new ones, tend to be biological indicators. And we've pulled out 4, 2 that are sort of, they're all related to soil organic matter, but they're sort of just pieces of that soil organic matter pie. Instead of looking at all of the soil organic matter that's present, let's just look at the organic carbon, and maybe the carbon that's easily available for microbes to break down. So the 2 carbon measures that we've pulled out are active carbon, or POC, and mineralizable carbon. Then for nitrogen, we've pulled out autoclave citrate extractable protein and potentially mineralizable nitrogen, or PMN. And I'll go through all 4 of these. I do want to note, we aren't saying that these other ones aren't important. If it wasn't important, I wouldn't have included it on this slide. But these are just the 4 that we've pulled out for the soil health study that we're conducting. All right, so some labile carbon measures. And labile just means that they're sort of readily available to be broken down, easily changed. Active carbon is a chemical test. And it does have a pretty widely accepted protocol. It uses permanganate to oxidize that sort of on its way to being stable organic matter carbon. So this is one fairly widely accepted soil health indicator that's maybe offers a little more information than just soil organic matter. It's a little more sensitive to management. Soil respiration is another one that's also, since it's not a chemical test, this is actually an incubation, it gets at the activity level of the microbial community as well, which is helpful and interesting, but it also comes with some challenges. So there are some pretty big variations in protocol, mainly in the length of incubation. So there's a 1-hour respiration, there's 1-day respiration, there's 4-day respiration. So this is something to keep in mind when if you are looking at labs to try to do soil health testing, which one of these are they running? And you don't necessarily want to compare data from 2 different length incubations because it won't make sense. All right, moving on to nitrogen. I have 2 measures for this as well. So soil protein, or autoclave citrate extractable nitrogen, is one of them. And soil protein is, or protein in general in soil, is sort of the biggest pool of organic nitrogen, that's in organic matter. So that's why quantifying soil protein is a great way to try to look at what might be available soon in terms of organic nitrogen. But this is a chemical test, just like POC is a chemical test, and it does have a pretty widely accepted protocol, which is great just in terms of sort of reliability of the numbers and comparability between labs. Next, we have in-season available nitrogen, or potentially mineralizable nitrogen. So the idea here is that this is nitrogen that microbes in the soil are likely to make available to plants throughout the season. And again, just like MinC, it's an incubation. So it's using the microbes that are present in the soil. And again, like MinC, it has some pretty big variations in the protocol. The big 2 differences are aerobic, so there's oxygen present, versus anaerobic, no oxygen present in this incubation. We use the anaerobic test because, so no oxygen present, because it's a little bit more logistically feasible. This is a 7-day long incubation, and the aerobic incubations are 28 days or 107 days. So that's just, it's a big ask for labs to potentially scale up to. Even 7 days is a pretty long incubation for sort of big soil testing labs to invest in scaling that kind of a test up. One con of using this anaerobic test is that the microbes that are in your field breaking down organic nitrogen are likely aerobic microbes, not anaerobic microbes. So that component becomes a little bit decoupled from reality in using the anaerobic one. So these 4 tests we think are potential indicators for farmers to use because they're a little bit more cost effective, logistically feasible. And by logistically feasible, I'm really talking about the fact that they can be conducted on dried ground samples and stored samples. So they do have to be dried under 100 degrees Fahrenheit. It can't be oven dried in order to allow those microbial communities to survive. But that is part of it. And then that they're sensitive to management. And that's kind of the goal, is to be able to track soil health improvements using something that's a little less coarse than, say, full-on organic matter percent. So these are the 4 objectives, 4 tests that we're using in the soil health and soybean system study that you might have seen sort of widely advertised over Twitter, and if you were at WABA last week as well. And these are a simplified version of our 2 objectives. So first of all, trying to connect management to soil health indicators. So using these 4 things as the response variable. And then secondly, trying to connect these 4 things to soybean yield, seeing whether or not soybean really cares about these soil health indicator values. There are lots of reasons why soil health matters. Healthy soils do tend to produce higher yields, but that's usually because of maybe increased water availability or things like that. Corn tends to be more closely coupled to nitrogen measures because it's a good predictor of how much nitrogen is going to be available throughout the season. And as most of you know, soybean is a legume, so soybean doesn't care quite as much about the nitrogen. All right, but moving on to a little more details about the study, I'd love to give a huge shout out to any participants. I saw names trickling in at the beginning. There are a ton of people on this video call right now that participated in 2020. And I was so thankful that we had to shift to having participants mail us samples instead of going out to sample. And everyone was really willing to adapt, and it worked out really well. Due to the huge diversity of soil types across Wisconsin, we need a lot more samples in 2021. So if you are interested in participating in the study, I will put up my contact info on the last slide. So far, we saw a pretty big spread of indicator values. I just have data for the 2 tests that we did in Wisconsin, so organic matter, loss on ignition, and PMN as well. But the interesting thing that we can see here, so this is split out by soil type. So sand, loamy sand, sandy loam, silt loam, and silty clay loam. So it sort of goes to decreasing sand as you go across. And then there's unknown, or just a couple of sites that I don't have the GPS locations for yet. But these are based on the Web Soil Survey as well. We didn't actually measure texture. And I will draw your attention to the fact that silt loam, has way more samples than the other one. So this is 138 samples, while the rest are under 20. But generally, we saw that with less sand, that soil is just more capable of holding on to organic matter, which is a relationship that we already knew. But part of the reason I wanted to show you this is that there is still a pretty big spread in these values, even within these texture classes. The red triangles are means, but these lines are medians, and these are box and whisker plots. But then with PMN, we saw a similar relationship, but actually everything besides sand was considered similar. That's why they all have an A next to them. So what I'm hoping for is that soil texture doesn't matter as much, besides maybe the really sandiest samples, and that a lot of these differences could be explained by management. For example, I might expect higher PMN in fields that have seen manure. All right, so to close out, I'd love to give just some tips for soil health testing on your farm. I would recommend using soil health indicators to track change over time. There is quite a bit of variability in them, and I don't think they're as useful for just a single snapshot. Next, pick a time of year to sample and stick with it, especially with the sort of incubations that use the microbial community present in the sample. You're going to see quite a bit of difference between different times of the year. If you did want to do fall and pre-plant, great, but try to stick with comparing fall to fall and spring to spring. Next, pick a lab and stick with it. I mentioned the variability and differences in protocol. So by sticking with one lab, you're more likely to sort of see that consistency. And finally, keep managing for soil health, even if you aren't testing for it. If you aren't ready to sort of invest in testing for soil health, I think that's totally fine, but that you can absolutely manage for this without necessarily testing for it. So with that, I'd love to give a huge thank you to our funding sources, as well as all of the collaborators and the Bean team and a couple of folks in the soils department that have made this possible. And in the yellow box right here is my contact info if you are interested in participating. I will drop that in the chat as well.
All right, well, that was a great talk, Lindsay. Thank you so much. I do not have any questions right now coming into the chat. But if more come up while we're listening to Shawn's presentation, I'll be sure to bring them up at the end because you'll be sticking around the whole evening. Any questions come up on your chat? I know sometimes people send them in the private line. Oh, we have one right here. Do you have any information on in-field spatial variability of the soil health test, trafficked soil or no traffic?
What, can you read the end of it really quick?
Trafficked soil or no traffic? I assume that's all good.
Yeah. So I don't have a good answer for the end portion. But for the beginning, we are looking at that as well and trying to just sort of assess how they might vary across soils in Wisconsin. But this has been looked at in Ohio, and the basic idea is that they tend to track similarly to organic matter. So as organic matter is variable across your field, so it might vary quite a bit across a big field that has different management zones, or especially with moisture. So increasing moisture tends to lead to increased organic matter because there's less mineralization going on. So that's one factor where it might change across a field. If you have a wet portion of a field, it might have higher values. But in general, they track very similarly to organic matter. And I think POXI and ACE track similarly to other routine soil tests, P&K.
Yeah, thank you. And we have one more question for you right now. Have you looked at plant parasitic nematode populations in terms of these soil health parameters?
No, I have not.
All right. Well, with that, I think it's time to pass everything. How was yield related to these measures?
Oh, that's a great question. And it's something that we're trying to figure out. but so far we need to do more research to see how soybean is related to these measures. It does seem to be corn is generally positively related to a lot of these, but and specifically the nitrogen measures. But also it tends to be that more organic matter might mean that corn is more it's like an increased capability of yield, but it doesn't, the resources also have to be there.
Interesting stuff. Well, I'm going to pass it on to Shawn, and if any more questions come in for Lindsay or Shawn throughout the next half hour, I don't actually know how long Shawn's talk is, I will be sure to pass those on to our speakers at the end. And just a reminder that we're going to save CCA numbers until the very end of both talks. Shawn will list the QR code for CCAs at the end of today's presentation, and at that time, if you're not going to use the app, you're welcome to put your name and your CCA number in the chat at the end of the presentation today. Thank you very much.
Awesome. Thank you, Emma. Thank you, Lindsay. We always want to thank the Fabulous Farm Babe for kicking things off, and thank you, Todd, for running quality control in the background. So again, for those of you that might not know me, my name is Shawn Conley. I'm the State Soybean and Small Grain Specialist at the University of Wisconsin-Madison. And today I want to kind of talk about some of the work that we've been doing on farm, some of the big data work that we have, and some things that are coming down the pipeline. So again, typical Conley style, I pushed basically an hour's worth of presentation into 20 minutes. So I will talk fast, but just know that everything that we sit here and talk about is also on my webpage. So again, my webpage is www.coolbean.info. Every time we write a research paper, we put out a publication. It's linked here. If you can kind of go through and see here, we have kind of separated out by soybean research, small grains research. And I always push things out on a listserv that growers and CCAs can automatically sign up for, as well as my Twitter page, which is @BadgerBean. So again, I think that's one of the things that we really try to do in the Bean team is push this information out and make it relevant to growers and make it relevant to the CCAs that support the farmers across the state of Wisconsin and, for that matter, the Midwest. So one of the things that we did is we managed the Wisconsin Soybean Yield Contest. And this is a yield contest that is basically across the entire state. And how we parsed it out is that we take, on a rolling 10-year average, we parsed it out into 4 different districts so that farmers are competing against farmers with similar type yields, plus or minus 5 bushels. Again, so we're not putting Spooner growers against, Iowa County farmers. So we have a little bit more of a regionality. And what we're going to do is kind of go through and break this down. This is the first, it's been published, and I will push this out on my Twitter page, and I'll push this out on my listserv as soon as this program is over. So how we'll do is we'll go by division, and then we'll go, and I'll announce second place first and then first place second within each division. So Division 1, second place, we have Jim Wilson. He's from Polk County, and he grew Asgrow AG 10X9 with an average yield of 71.74 bushels. So congratulations, Jim. And I should make the point that second place within each division gets a $500 check and a plaque, and first place gets a $1,000 check and a plaque. So those, I believe, I think Adam Roth, one of my technicians who helps manage and support the Soybean Yield Contest, has already put those out in UPS and FedEx, so they should be showing up in the next day or two. So with that, and then in Division 1, first place was Paul Graff from Door County. He planted NK brand S14 U9X and 71.96. So again, you can see how tight this is. We double checked the math, and there was only 0.22 bushels difference between a $500 check and a $1,000 check. But again, this has been probably the best number of entries we've had. I guess COVID is good for one thing or another. At least we had people interested in actively pursuing and entering the Soybean Yield Contest. Next, in Division, Division 2, we have Mike and Dean Wegner from Monroe County. They planted Pioneer P23A15X with a yield of 81.32 bushels. That means for the first place in Division 2 would be Paul, and forgive me, I'm going to butcher this, Lapasinski from Barron County, and he planted another Pioneer brand, P16A13X at 82.14. Again, you can see how close it was this year from the entries across the state. So it was actually kind of a fun year, and we had to do a lot of checking math and making sure everything was right. So we announced the farmers accordingly. In Division 3, in second place, Ryan Bates from Pierce County. He planted Pioneer P16A84X with a yield of 80.6 bushels per acre. Then in Division 3, first place was Tim Grafton from Kenosha. Planted Pioneer A80X at 90.22 bushels per acre. So again, really good yields across the state of Wisconsin. And I would say that in speaking to all the farmers that I work with, you know, from Spooner South, 2020 was a very good growing season. And for the most part, most of the farmers would classify it as a top 3 growing year for their own home farm in terms of soybean production. And if you look, we planted roughly 2 million acres in the state, and we averaged 53 bushels per acre. So again, we might not have had the record numbers in terms of yield, but we were pretty high as record in terms of total production. So it was a very good year for most of the farmers. I do know there were some dry spots that came in around in August and early September that probably took 4 to 5 bushels off the top end. But again, for the most part, a good yield. All right, now let's go to Division 4. In fourth place, not fourth place, in second place, Ron Digman from Grant County planted Pioneer P28A42X and averaged 98.29 bushels per acre. So getting close to that 100 bushel mark. Well, in fact, our first place winner, Don and Doug Mipton from Dane County, grew Asgrow AG20X9 and hit the 105.18 bushels per acre. So this is the first time that we've actually had farmer cooperators, participants hit that number. The UW Bean Team graduate students hit that number about 3 or 4 years ago, I believe. But again, they were not able to take the check. And for the first farmer to surpass the 100 bushel mark, that's an extra $2,500. So Don and Doug, hopefully you're here. I know we talked earlier to get you on. Congratulations, it's $1,000 for first place and another $2,500 for hitting that 100 bushel plus mark. And if we were at Corn Soy Expo right now, I would be asking you to buy me a drink. And if I see you, I probably will still do the same thing. But you know, yield is obviously important, but we also have to look at quality. So we actually have a quality test as well. And that winner was Rick DeVoe from Greene County. He planted Pioneer P28A42X. And what we do is we take the yield, his protein content and oil content, and we look at that. Rick harvested over 3,000 pounds of protein and oil on a per acre basis. So that's awesome to know as well. So again, just want to take this time and thank all the winners, have everyone look forward to putting out plots in 2021. And look forward to this stuff getting posted on my Twitter and sent out on my listserv here right after this presentation is over. All right, so to jump back into this, just want to remind people about the growing season we said. Earlier, I talked about, you know, how we had record production and near record yields across the state, and just so farmers understand how the range would go. So we're at Spooner, where we average 56 bushels per acre. Where we get down here to Platteville, Janesville, and Arlington, we're at that between 78 and 86 bushels per acre. And we had some high yielding varieties at both Arlington and Platteville, average in the 90s. So again, very good production year for many farmers across the state of Wisconsin. Now, one of the good things about running the variety trial across the state of Wisconsin is we being able to look and analyze these traits as they come in the marketplace. So how you read this is obviously this is yield on the y-axis. On the x-axis, what we're looking at is the different regions. So combined regions would be the entire state of Wisconsin. Then we have the southern, central, north central, and north regions. The blue bar is would be the E3 trait. The orange bar, that's what at least it looks like to me, the color would be LLGT27. and the gray bar is the Roundup Ready 2 Extends. So if we look across the state of Wisconsin and average all of these different areas or regions, we see that the LLGT27s and the Roundup Ready 2 Extends were both similar in yield and outperformed on average all the E3s. However, we're only talking about 1.5 bushels to 2 bushels on average. If we start breaking it out into districts, for example, the southern region, we saw E3s outperform on average with all the varieties that we tested, the Roundup Ready 2 Extend. So again, the point I'm making here is that there are very good varieties in many different platforms and farmers have options. And that's really the point. I really encourage you all to dig into our variety testing trial, get a sense and look at how these yields perform on a specific variety basis and be able to choose those traits or those platforms that best fit your growing season and how that best performs in your area. So again, that's the point of this. So go to my website, coolbean.info, and go and download our variety testing book, and you'll be able to look at the variety performance based on the genetics as well as the different trait platforms as well. So as Lindsay had said earlier, one of the challenges we have in Wisconsin is getting cover crops established early enough that we can get a high enough yield to be able to produce enough biomass out there to really matter. So what we did is we basically took all of the varieties entered in our northern district and we planted them down in southern Wisconsin at our Arlington site. So typically at our Arlington site, most of the varieties are mid to late 2s that we're looking at. So here we're putting anywhere from a 0.4 maturity group bean all the way up to a 1.5 maturity group bean. And if you look on the right-hand side, we see very high yields. On average, if you looked at how these early beans did against late beans, I think there's on average 2 to 4 bushel yield difference. So again, the point I'm making here is there are some opportunities, I think, for farmers that want to establish cover crops on some marginal land to be able to get an early enough maturity group bean planted, i.e. planted it early, because all of our beans are planted by May 8th this year, and be able to get those out in a timely manner to get your cover crop established. And John Gaska, one of my research technicians, also helped put this together. So how you would read this is this would be our recorded maturity date, i.e. when those soybeans hit R8 growth stage, okay? These are our relative maturity beans all the way down here from like a 0.2 maturity group bean through our early maturity group test. And then we jump into our late maturity group test that took them all the way out to a 2.8 and a 2.9, all right? So these dots are the individual varieties and what date they hit R8. Then on average, what we see is roughly a 7 to 10 days after they hit R8 is when you can physically combine them and then plant right afterwards. So again, this gives you an estimated, in this case, we're talking winter wheats, but it can be cover crops as well. So again, if we look here, we have some early beans that can come off that yield well. And again, this isn't yield per se, but they yield well, come off, and we get them established in a timely manner. So again, what we saw here is basically for every one relative maturity group, so I mean from like a 0.5 to a 1.5 maturity group bean, we saw 15 days delay in when they hit R8 or when you could physically go out there, harvest them, and establish either a winter wheat or cover crop. So again, this information we just posted on our webpage. Feel free to go in and talk about this. Is basically talking about how to establish early maturity group beans and then get your winter wheat or cover crops planted on time. So again, I think there's opportunities for farmers and to be able to look at this. All right, now let's kind of get into some of the talk about our on-farm data. And many of you remember, this was probably 5 to 6 years ago, we started talking about this whole idea about big data and the importance and value of big data. And, you know, we've been hearing about this for, well, at least a decade. With today's computing power, the ability for our electronics or equipment, we can go in and be able to how to utilize this big data. And that's what we're really trying to capture. Farmers are sitting on a pile of data. How can you best use it to predict what management practices you should be doing moving forward? So The first part of this project that was funded by the North Central Soybean Research Program was called the benchmarking project. And if you see here, this was 4 years of yield data from across the North Central region. We got over 8,000 farm fields and over 600,000 acres of data. Pretty much this is the biggest soybean database out there in the public sector that has talking about soybean practices, soybean yield, all right? So again, this is the data set that we're working with. Some of the early data, and I know I've showed this quite a bit, is we're looking at some of the simple or the main effects, I would say, and a big one on soybean would be planting date. So I'll just pick this orange coloration here, that would be 5R. So if you look on this Wisconsin, Northern Illinois, this would be Eastern Iowa, this orange coloration, that's TED 5R. And we see that basically from April 25th, all right, we're losing 0.4 bushels per acre per day by delaying our planting date. So basically going from April 25th to May 5th, if I did my math right, that's 10 days, we lost 4 bushels by delaying our planting those 10 days. So again, this gives us an opportunity to really look and hone in, because in some of these other areas, let's say in this 2R area that covers part of Indiana, Ohio, parts of Illinois, they're only losing 0.15 bushels per acre per day. So again, maybe it's not as the need isn't as strong in some of these locations to get in and plant early. All right, so this is one of the frameworks that we're looking at, this large data set. But the other part we want to look at is how can we utilize all this data to kind of have a systems approach. Now, I'm only going to be speaking about 2019 data because we finally just got all of our 2020 yield data off of yield monitors, and my goal is to have this report posted on Cool Bean on February 1st. So I think we can make that happen. But these are what we were doing, is looking at our interactions and going into these specific TEDs and taking data and information we gathered from our models and making basically system-wide predictions of what farmers can do to minimize that yield gap on their farm. All right. So in 2019, we had 49 replicated trials across the North Central region. If you look at the power and the impact of this experiment, we're sitting at covering roughly 30% of the North Central region, which is roughly 80 to 82% of all U.S. soybean production. So this is a big chunk of soybean acres. We're really trying to bury down and trying to figure out what farmers can do on their farm to minimize the yield gap and increase yield. So this is kind of an idea of one of the experiments. And this is one of the reference forums from Nebraska. And what we did is we worked with farmers. We gave them their reference, which the reference trial would be, what do they normally do? What are their normal practices? And then we provided them an improved practice that they can implement on their farm to either increase yield or cut input costs. So that's our comparison, improved versus a reference. So again, this is more of a systems trial, which is kind of hard for some agronomists to come into, because we usually have these big factorials and we're trying to parse out, which factor gave us what. And on these on-farm trials, it's really difficult given the number of treatment differences that we're seeing. So that's kind of a framework. And again, this is posted on Cool Bean. If you want more information about it, that report is there as well. It's a boots on the ground report. So if you look across here, these are the different treatments that we're looking at. I'll just point out Wisconsin because we're living in Wisconsin. Basically, this is saying plant your beans if the soil is fit up to 2 weeks earlier, and drop your seeding rate down around 130,000 seeds per acre. Okay, and most farmers in Wisconsin are pushing that 155 to 160, and they're planting, roughly on average May 10th. So we're really trying to push farmers plant early and drop their seeding rate. And we see different interactions for Ohio, not just interactions, but treatment profiles, Minnesota and North Dakota. All right, so let's get into skinny. So how you would read this figure right here is this is the yield on the x-axis of the reference. So basically what farmers are doing on their farm, okay? This is the yield of the improved treatment, what we suggested based on our data analytics that would increase yield and or decrease the amount of input costs associated with that. This red line right here is the one-to-one relationship. So what that means is if a field, in this case, there's a field in TED 6, fell right on that, it really didn't matter in terms of yield. Okay, we'll talk about economics a little bit later, how it fell out. But what we see right here, we see a lot of fields well above the one-to-one line, well above the 5 bushel, and several above 10 bushel in comparing the improved treatment versus the reference. And in fact, on average, we roughly hit on the 47 paired experiments on farm trials, almost a 6 bushel per acre yield increase. All right, so that's cool yield. Now let's talk about economics. So with that being said, I'm going to show you how you read this figure. So this is a percentage of overall farms. So again, we're looking at those 47 farms that we got all the data for. This is the additional profit derived from the improved treatment. So this is a 0 line. So on the 0 line would say that whatever you did on the improved gave you the same profit that you did on the normal or the reference treatment. Now, if we look here, this dashed line would be $10 per acre more. Look at all these fields that are well above that. And in fact, 85% of the cases had extra profit greater than $10 an acre in our improved treatment versus the reference treatment. And on average, again, we're looking at $55 per acre more cash in a farmer's pocket by implementing the improved treatment over the reference treatment. So again, we'll be doing this and combining data from 2020. We're going to be doing this again in 2021. So if anyone's interested, feel free to contact John Gaska. I don't know if you all can see this, but it's jmgaska@wisc.edu. Emma, can you type that into the chat? If anyone's interested in doing one of these on-farm treatments, we'll be glad and happy to work with you and kind of share that as we go forward. All right, how much time do I got left? Got 10 minutes, I can do it. All right, the last thing I want to talk about is a kind of a cool project that we've been working on for several years. And what we want to do is advance not just our agricultural research, but on-farm management decisions using AI algorithms. Basically, putting all this data into a model and be able to predict on a per acre basis or a field basis what farmers should be doing in terms of genetics, management, and to improve not just yield, but profitability. So I'm going to talk a little bit about this. So this is, I don't want to say it's totally pie in the sky. We've gotten a little bit of traction on this. We're starting to work on it, but I thought the farmers here would be interested in seeing where we're at with this. So these are the questions we get from farmers. You, Shawn Conley, you do all these different trials and you're looking at 1 or 2 practice separately, okay? So you're looking at seeding rate and you're looking at row spacing separately or genetics by seed rate treatment, okay? So we really want to understand how can we optimize this more than just on a separate basis. So how do we test these interactions? We have like I said earlier, row spacing by seeding rate interaction, okay? How does that affect yield? How about a 3-way interaction when we implement planting data on top of that? How about we look at the entire cropping system? And to tell you the truth, it would be physically impossible. I don't have enough money in my budget, and I don't think any other person or entity, for that matter, would be able to go and look at 10-way interactions to maximize yield. So we have pieces of the puzzle. All right, how can we effectively put them together to tell us a story and inform us about how we could be looking at and maximizing and optimizing these different interactions? All right, so how are we going to do this? All right, here we go. So what we want to do is develop a method that allows us to rapidly and accurately predict environment-specific interactions. Okay, these are complex interactions that are going on out there, G by E by M. But that data is really hard to come by. So what we've done is we've basically gone through and gleaned every single bit of publicly available corn and soybean yield data, management data, weather data, and then we're trying to leverage the power of machine learning algorithms to basically predict and evaluate thousands of potential cropping systems and identify which one of those would best work on your piece of dirt, if you will. Hopefully there's no soil scientists out here to shame me for saying the word dirt when it should be soil. All right, so this is the data that's in our database right now. So basically for corn, and shh, don't tell Lauer, I'm working on corn, all right? So on corn, we got roughly 17,000 G, genetics, E, environment, and M, which is management specific yields. On the soybean side, we have roughly almost 25,000 G by E by M specific yields. Okay, and we looked across the entire US for this project. So just for a frame of reference, the yellow is primarily where corn is grown. The brown is where soybean is primarily grown, just so we kind of get an idea. This is where we collected yield data from. Any red pin would be soybean, and any gray pin would be maize. We wrote this for a scientific journal. Corn is what we're talking about here. So you'll see that we're obviously most of the data came from the Midwest, but we're able to kind of tease and capture data from all parts of the United States that grow these different crops. So again, I'm not going to talk too much about this. I just want to say that, yeah, we're working on the stats. There's a lot of math in the background. And again, I think, and Adam can help remind me, but I think it took us over 2 years to build this database. So this has been a yo person's, it's been a lot of work. And I'd like to thank all the undergrads and graduate students that have done this, because it's a lot of coding, a lot of time going in and gleaning data from all these different websites across the United States. So this basically here, I'm not gonna spend a lot of time. This basically just tells you, again, it's the one-to-one relationship, the difference between actual and predicted. And we've gotten our error down pretty close. So what that means is our error is within 7.8 bushels on the corn side and 3 bushels on the soybean side. So that's pretty good. I mean, that's better than most LSDs you will see on any type of a published manuscript. So again, it's a lot of time, a lot of effort, and I think the models are pretty good. Here are some of the results I want to kind of share with you. So again, the top panels, A, B, C, D, all right, that would be corn. The bottom panels, E, F, G, H is soybean. So how you would read this is a mean centered yield, or what's the average yield, okay, based on this different planting date. So this is the 80th day of the year. So remember, we're doing some stuff in Texas as well. So we show on the corn side, we see as planting date is delayed a little bit, we hit a peak right around that 100 and I don't know what that would be, 110 days. And then we start seeing the planting date response of corn go down. On the soybean side of things, we see very flat, and that's pretty similar to what we see. Is, for the most part, it doesn't matter if we're planting in, March 20th in Indiana or April 27th in Indiana, that yield response is the same. However, we don't have any December 10th data in this, so that might just muck up the system. That's kind of a joke for some in the audience. I don't know if he's here or not, that actually planted his beans on December 10th in Wisconsin, and he dug them up today and they were still frozen. So just want to give him a shout out for trying something crazy out there. We also see seeding rate, nitrogen rate, row spacing. Again, I don't want to spend a lot of time on this, just showing you all the interactions we can look at. So what we did is we ran a hypothetical scenario, all right? So we looked at, just picked out 2 days. We picked May 1 and June 1, okay? Some of the different input variables we have in our algorithm is conventional till versus no-till. Different seeding rates. We have a whole string of seeding rates. We just picked 2, 140 and 160,000. Row spacings of 15 and 30. Did we use a foliar fungicide? Yes or no? What maturity group did you use? And what was the previous crop? Again, we can do this for a lot of different interactions. I just want to give you all an example of how this algorithm works. So what we're really looking at At Arlington, Wisconsin, we were looking at 128 different management combinations over 5 years, which gave us 640,000 G by E by M specific yields. So that's what we're really kind of teasing out, is looking at the genetics. All right. What traits? And we didn't talk about that here, but we have the traits in there. The environment, obviously the weather changes, and the management, and that gets into seeding rate, row spacing, foliar fungicide, etc. So I'm going to walk you through this. All right. So here's the effect of planting date on average. So this is just looking at planting date by itself. Just one factor, like we were doing a normal experiment, just looking at planting date. And what we see here, the difference between a May 1 planting date and a June 1 planting date is 14% yield difference. All right. Again, forgive me for the metric system, but this is in review at a manuscript, and I'm going to wait till it gets accepted before I turn it into English units. All right, next. So this year is a difference due to 128 different cropping systems, all right, or 640 specific yields. So we see here the difference on average is 15%, but you look at how big the variability is, as shown by these box and whisker graphs that Lindsay showed earlier, very similar. And this is the yield difference of the whole cropping system. From top to bottom, we see a 44% yield difference. All right, that just shows the variability. So we can really hone in on a land-specific point and estimate yield on which management practices farmers should be doing. And based on a specific cropping system, here when we're only targeting early planting systems, we still have 27% variability, which tells us that even within an early planted system, there's a 27% yield variance around what we can produce. So again, I know this is crazy math and way out there, but I think that's something that we can have going forward. So what we can then do, I forgot about this one, is that we can basically tell you as a farmer what is the highest yielding system on your land. Not that you'd want to know the lowest yielding system, but we can identify that as well. So again, that's more of a fun tool that we can play with, is in that yield variation. So with that, there's my website, coolbean.info, Twitter on BadgerBean. The soyreport.blogspot is where I'll be posting this data in terms of the yield contest winners. And with that, there is your QR code, and I am right on 7:59. So
Well
with that,
done.
I'm going to hold this up for a couple minutes so people can snap their QR codes for CCAs. And if you could please, if you don't have the app, type in your CCA number into chat so that we can capture that and you can get credit. So with that, I'm going to pass it over to Emma.
Yeah, I'm also reading through the chat as you drop CCA numbers so that I can catch any questions for Shawn there. So if you have any questions, put them in the chat. But I really enjoyed your talk, Shawn. Great math with those scarlet and gray pins. Huge fan. And I'm just waiting for some questions to come into the chat.
And I love how you're proud of yourself for finishing right on time and leaving no room for Q&A.
Hey, that's pretty
so
good.
Lindsay has a question for you, Shawn. How far away do you think this algorithm project is from on-farm use?
So we actually did get funding from the Wisconsin Corn Growers Association to test it in corn in 2021. And I put in a proposal to the Wisconsin Soybean Marketing Board to test it on farm as well. And I'm just waiting to hear the results on that. So I'm guessing, widespread application, it's probably 5-8 year more, I don't know, down the road, but we're going to be testing it this year to see if we can break it.
Nice. That was sooner than I expected. We've got another question here for you. Will there be any considerations of fertility programs in the modeling efforts?
Yes, we do have some fertility components in there. Again, we're using public data, and not all research specialists are as good as mine and report fertility levels and how much fertilizer was applied in every report they do. So yes, we have that in there, like nitrogen rate on corn. We have that. We have P and K applications, but they're not as robust as others. But with that being said, Emma Matcham is doing a lot of stuff on the soil fertility side, trying to look at rethinking how we do some critical levels on the K side. So stay tuned for Emma. I'll have her up next year doing, hopefully this will be live at the Kalahari so that I can have people that win the yield contest buy me booze with their thousands of dollars versus drinking a bottle of water at the Soybean Association office. So.
We've got one more question on the topic of P and K. In the top yielding soys, where is the P and K value?
Are you talking about what the yield contest? I guess we have the information if the farmers provided it for the yield contest. I mean, again, if we could dig in if we're talking about the algorithm, I have that. We just didn't have that in this iteration of how we ran that. Regarding, I guess that kind of really gets into actually Emma's PhD project where we're doing sampling and trying to estimate variability. Well, hell, you just tell them what you're doing, Emma. You probably know it better than I do.
I'm working on a database project where I have farm management history and past yield maps on around 700 fields in southern Wisconsin. And we're seeing how closely the yield and soil test potassium are related to each other and whether that relationship changes over soil texture, elevation, other field properties, so that we might be able to, in the future, evaluate critical levels that change based on other relevant soil properties. And right now it looks like there's not a relationship between soil test K and yield once you get over the critical level for your given soil texture in Wisconsin. Shawn, I have one more question for you right here that I want to make sure we get to since we're 3 minutes over time.
Okay.
Early planting date is great, but is there a minimum soil temperature for us in the far north?
So yeah, good question with the soil temperature. So a lot of this fundamentals work, physiology work was done in the 70s. And in the 70s, we planted soybeans in June and they didn't really care about what the base temperature was. They figured it out for corn and said, hey, it's 50 for corn. Let's just say, hey, it's 50 for soybean. There's a lot of debate out there. I mean, it's less than 50. It's, I don't know if you want to call it 44, 46.5, I do know you can have inhibitional chilling in soybean, but it's not as, the window to get it is a lot shorter than it would be for corn. We've planted beans in Hancock with no organic matter on April 28th, and it froze the top quarter-inch of the soil, and they came up and emerged just fine. So again, I think we don't give soybean seed enough credit. Obviously, you have to look at the vigor ratings for those seed lots, but so as a collective group, what we're suggesting is you look at what the crop insurance date is for your specific county, and that's when you start planting. For most of Wisconsin, is somewhere between April 20th and April 25th. That is our recommendation. But if it's April 15th, I'm in central Wisconsin and the soil is fit, I'm going to let her rip. I know that's really sciencey, but that's what I would do.
And we actually have a few more questions. Are we okay going a little over time tonight? I don't know.
Yeah, the call is set up. We're good to go as long as people want to ask questions and our two hosts want to continue to answer them. We're good.
I'm good. Let's go.
All right. And shout out to Scott, who asked our last question, for managing to type a really excellent Northern Wisconsin accent that I'm not going to try to emulate as an Ohioan.
Please
not going to happen.
the question appropriately.
Does the M in your G by E by M studies take into account disease control management? And that is from Mitchell.
Yes, they do. So if we look at that's plus or minus foliar fungicide, plus or minus insecticide, again, all of those factors are taken into account.
And for clarity, that's looking at whether the pesticides are applied, not looking at disease pressure. So we're not
Correct.
trying to model the disease pressure itself.
No, that's a different project we're working on, but that's something we're working on with Paul Easker from remember that data set I showed earlier? We had 8,000 farm fields and 600,000 acres of data. We're looking at epidemiology modeling from that data set, but that is not in this one. This is just basically prophylactic applications at R3. This is what we did. This is what it yielded.
All right, and then I have a question from Bill. How long before we have a usable model to predict in-field yield variability? As somebody that works with in-field yield variation and big data, I'm gonna say a long time, but I'm curious to hear what you have to say, Shawn.
So I'm really good at predicting yield. I start with 50 bushels per acre, and it's plus or minus 50 bushels per acre, and I'd get it right about 98% of the time, except for the one farmer that had over 105, and I would have missed that one. Yeah, it's really tough. Soybean is, because it's such a phenotypic plastic plant, I think we'd probably be closer on the field corn side of things, because it'd be easier to model corn. I mean, corn's pretty easy. Soybean is really a challenge to work with. That's where the real scientists work, is on the soybean side of things.
And not to step on your toes here, Shawn, but there's some really good work from Michigan coming out about in-field yield variability. And what they're finding is that there are some spots in your field that you're going to be able to predict, and there will be some spots in your field that no matter how many years of yield maps you have, they will have unstable yield trends just due to that environment and the growing season changing year to year. So there will be some spots that we cannot predict before we see what that season brings. But other spots we can predict with a few years of yield maps.
Thanks, Emma. You're probably up on the literature more than I do.
Yeah, that's what you pay me for. And we've got a question here from Jerry. What pre-herbicides are easiest for early soybeans to metabolize?
About a month ago, we had a really good wheat scientist talk. So I'm actually going to balk on this one. I'm not as good on the herbicide, metabolism and cold temperatures. Rodrigo Worley is, and he'll be speaking. I don't think it's next week, but the week after, but he'll have some really good information on that. So I'm sorry to pass, but I don't want to give bad information. If you want to, can you email me at spconley@wisc.edu? Oh, there you go. Thank you, Haley. She put up his website for Rodrigo. Either drop me an email or go to that website, and then we can help you out with that one. But whatever I would say here would probably be wrong, and I don't want to say it.
And I think that's all we have for you today, Shawn. I'm sorry the last two questions were a little far outside your wheelhouse, but we appreciated your meme. We appreciated your talk.
I was hoping someone would catch my meme that I had hidden in there.
Oh, well, we did get a question in the chat from John. Is Bernie in Ryan's field? He's dressed for it.
Exactly.
And that's all I have for you today. Todd, do you have any closing remarks about next time, or not to put you on the spot, but to put you on the spot?
No, I was quickly looking at the schedule to figure out when Rodrigo is speaking. It is not either of the next 2 weeks, but on badgerbean.com, you can look at the schedule. Registration's open. Apologies for the people that had problems joining early. We made sure Pam went a little longer to give you a chance at that. So we just look forward to the next time. Thanks, Shawn. Thanks, Lindsay. We really appreciate your time tonight. Emma, great job as usual, and look forward to the next one. This is recorded, so we're gonna package up a handful of these and they'll be on our YouTube channel. So you'll be able to see Shawn's right away, and we'll make sure whatever materials they post to share, we'll share.
Awesome. Well, thank you, everyone. I want to thank everyone for taking their Thursday night, and hopefully you were all sipping on a cocktail and enjoying this. So we'll see you later. Thank you.
Thank you.